Welcome back to Mendelian Genetics. Today we are going to be talking about dihybrid crosses and how you can use them to figure out what a person with multiple traits can be. And if you can use a dihybrid cross, then you can figure out a trihybrid cross, and you can go anywhere up to even a cross that has 100. I don't see why you would want to, but you could. So that's what we're going to be working on today. So for a quick review of Mendelian genetics, Gregor Mendel is the father of genetics. He studied pea plants in Austria around the 1800s, where he noticed the two laws that he called them, the law of independent assortment and the law of segregation. And Mendelian genetics, as we call it, is one character coded by one set of alleles, one characteristic, one trait coded by one set of alleles. And all that means is you're either going to have it or you're going to show it or you're not going to show it. There is no in between. And that is Mendelian genetics. A monohybrid cross. This is a cross of one set of alleles using a Punnett square. So this is your big T, little t crossing with a big T, little t and figuring out what's in the middle. You get four squares, four possible children, and that's what we see. Homozygous dominant. Well, homozygous means the same allele. If it is dominant, it means that that trait will mask the recessive trait. That trait will show if paired with the recessive. So homozygous dominant means that we're going to get both of that same dominant trait together. So big A, big A. Homozygous recessive, this is the same recessive or the alleles that will be masked if paired with the dominant. And the only way this recessive allele will show in Mendelian genetics is if it is homozygous. So example, little a, little a. And then the last but not least, heterozygous. Hetero meaning opposite. One of each allele, and the dominant trait will show that big A is going to mask the little a. So homozygous, homo, same. So homozygous dominant, big A, big A. Homozygous, homo, same. Homozygous recessive, little a, little a. And this is the only way that recessive trait is going to show. And then heterozygous, one of each allele, because hetero means opposite, and you get that big A, little a. So the Mendelian inheritance and the rules of probability. This says that the rule of multiplication, the probability that two events will occur simultaneously, is the product of their individual probabilities. So the probability that an egg from the F1 generation will receive the little p is 1/2 because we have the two alleles there. So it's 1/2 or 1/2. It's a 50% chance of getting either. The probability that a sperm from the F1 generation will receive that little p is also 1/2. So if we wanted to figure out the probability of receiving two alleles during fertilization to get that two recessive alleles, we get 1/2 times 1/2, which equals 1/4. The rule applying to dihybrid crosses. So for a dihybrid cross, we get a big Y, big R, little r. And that's going to say the same for both parents when we go to this particular example. And it's asking, what is the probability of an F2 having the genotype of big Y, little r, little r? Now, the biggest thing here when it comes to this one with the rule of multiplication is we are going to figure out what is the probability of each of these things having. So first things first, what is the probability of getting a big Y? Well, for the first one, it is 1/2. And then we're going to multiply it by the probability of getting another big Y, which is 1/2. And the same thing for the R's, 1/2 times 1/2. And that's going to be 2 times 2 is 4, times 2 is 8, times 2 is 16, 1/16. And we're going to show you how that works in just a little bit. So a dihybrid cross is made when the phenotypes and genotypes composed of two independent alleles are analyzed. And this is very similar to the monohybrid cross. So what happens is we get two traits being analyzed, which would be our plant height of tall. So I'm going to highlight these real quick for you. The plant height being tall with being heterozygous. So our tall being dominant to short. I'm going to underline that. Tall being dominant to short. And our flower color with a big W and a little w. And we're going to say for that one, the purple flowers being dominant to white, exactly how Mendel saw them. And we're going to create that dihybrid cross. And the first thing we're going to do is we're going to figure out how we're going to get these, alleles, because in a monohybrid cross, we know that one is going to get one T and the other one's going to get the other T. Well, it's slightly different when we go into higher dihybrid crosses, because what this is going to do is we need to get a T and a W and so on and so forth. So how we're going to do this is we're going to look at it through what we can do as the FOIL method. So we have a big T and a little t, a big W and a little w. And if we look at the FOIL method, FOIL is first, outer, inner, last from your math classes. So first thing we're going to do is we're going to look at the first two. So the first two, that's a big T and a big W. So we're going to put them down here, big T, big W. Our outer ones, so we're going to do this one in orange, outer, which is a big T and a little w. Our inner ones, so it's a little t and a big W. And then our last ones, and we're going to put this one in green, our little t and our little w. And it's going to be the same with our second one because our second one is the same as that one. So we're going to have a big T and a big W, a big T and a little w, a little t and a big W, and a little t and a little w. Now, when we go to solve this, we're going to solve it exactly like we would a monohybrid cross, but we're going to put the t's together and the w's together. So I'm going to do this in, we're going to do yellow. Let's see how yellow turns out. Nope, we can't do this in yellow. So we'll do this in a darker color. We'll put this in purple. So we have a big T, and our big T is going to go down the column. So we're going to put all these big T's down this column. And here in the second column, we have all big T's. So we're going to put, so the second column right here. And this all has big T's, so we're going to go down this column with big T's. Big T, big T, big T. Now we're going to go to the third column right here. And if we look, this one has little t's. And remember, whenever we have a big T, we want that big T to come first because it makes it easier to read. So we're going to put this little t in that second position just in case we have a big T coming up. And then we're going to look at this last column, this fourth column. And right here, once again, we have that little t, so we're going to put that in the second position just in case we get a big T popping up, because this second position makes it easy to read. Now, same thing with the W's, but let's worry about the T's first. So let's go to this first row, and this is a big T, so we're going to bring all these big T's across. Same over here, let's bring all these big T's across in the second column. And over here we have a little t, so the little t's are going to go into the second position, but here we have a little t already, so we're going to put this right here. And we're going to do these little t's here because this fourth column. And now we're going to go to the w's. Let's do the w's in a different color. We'll do the w's in orange. So first things first, this first column, we're going to put the w's, and all these w's are big W's. go to the second column. All these W's are big W's, and we're going to just fill these out. Third column, oh, wait, that was my bad. Hold on. These are all little W's. So that was me thinking ahead of myself. So here we have little w's, so we want that little w to go in that second position because that second position is going to make it easier for us to read. Now this one has a big W, so we're going to put all these in that, all these big W's down. And over here we have little w's, so we're going to put all these little w's right here on that second position just to make life easy. And then right now we're going to go across on all of them. This is a big W over here, a big W over here, big W over here. And here we have little w's. And here we have a big W. Like I said, you really want to make sure when you do these, you make your letters very different. And so on this last one, we have other little w's. So we're going to put these little w's down over here. Now, let's count up our phenotypic ratio. Remember, if we see, and we're going to circle, we're going to use yellow, green, dark blue, and light blue to circle this. So the first thing we're going to look at is if it shows the dominant in both, traits. And we're going to use the highlighter. So this first one is if it shows purple and if it is tall. So all of these that are purple and tall, I'm going to highlight. Oh, that doesn't work well. So this one's purple and tall because look, it has a big T and a big W. This one's purple and tall, big T, big W. This one's purple and tall. This one's purple and tall. This one's purple and tall. And this one's purple and tall. And this one and this one. So if we count those up, that's 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 are purple and tall. We'll go and circle in blue now. And they're going to do tall and white. So we'll circle all those that are tall and white. That's 3 that are tall and white. And let's circle all the ones that are now short and purple. 3 are short and purple. And our last one, we're going to circle all the ones that are short and white. Oh, that one doesn't work out so well, that color. We have 1 that is short and white. So that is a 9-3-3-1 ratio. 9 are purple and tall. 3 are purple and short. 3 are white and tall. And 1 is short and white. 9-3-3-1. It's really helpful to know that ratio when it comes to a dihybrid cross. In fact, I would really recommend memorizing this. Write it, star it, 9-3-3-1. And remember, this is a phenotypic ratio. That was a really bad handwriting there. My apologies. So that's a phenotypic ratio right here. Phenotypic. All right, I'm going to erase this real quick and we're going to move on. Now what we see here is exactly what we did on the last slide, But what we're seeing is them doing it, so it looks a lot nicer. It's not my handwriting. What you see is they have come up with the same alleles we did through the FOIL method, which we see a big T, big W, a big T, little w, a little t, big W, and a little t, little w. Now, real quick, before we begin, I'm going to write them down on the side. So we have a big T, and that means that it is tall. A little t, and that means it's short. A big W, and this means purple, so purple flowers. And a little w, and this means white flowers. So let's circle up what we see. So let's go for a tall and purple flowers. We're going to count them. That's 1, because that has a big T and a big W. And remember that little w is masked because it is recessive. 2, 3, 4, 5, 6, 7, 8, 9. So that's 9 that are tall and purple, though their genotypes may be different. Remember, we're looking at the phenotypic ratio right now because we're talking about what traits we see with our eyes. Now we're going to look at those that are tall but are white. And we have 1, 2, 3. And those are tall and white. Now let's look at the short and purple. We have 1, 2, 3. That's 3, that's short and purple. And last but not least, we're going to look at the last one, which is 1 by itself, and that is short and white. And so this is our 9 to 3 to 3 to 1. Star it, memorize it. This is going to help you so much with the phenotype. This is our phenotypic ratio. Now, this 9 to 3 to 3 to 1 is a phenotypic ratio. Very important to know. It's why I keep going back to it, which means that 9 out of 16 shows both dominant traits. 3 out of 16 and 3 out of 16 will show one of the recessive traits, and 1 out of 16 will show both recessive traits. And this indicates that the law of independent assortment is true because they are going to separate independently of the other trait. If that law was not true, we would have that purple and the white would be or the purple and the tall would somehow be connected, but they are independently assorting of each other, so this law is true. At least when it comes to Mendelian genetics. Next time we meet up, we will be talking about codominance and incomplete dominance, and we'll go over some more. And these are when we start going out of that Mendelian genetics, where we're going to start talking about how things like hair color and eye color and other things can start being affected, and how us as humans don't necessarily fit the same mold as pea plants. See you next time.